For centuries, medicine viewed your genetic code as an architectural blueprint. Once the house was built, the floor plan was locked in. If there was a flaw in the foundation, doctors could patch the cracks or reinforce the walls, but they could never redraw the original design. Within the human genome, there are roughly 20 to 25,000 individual genes. The margins for error are incredibly tight. One single spelling error in this vast sequence can trigger a biological cascade, resulting in severe, complex conditions like cystic fibrosis or Huntington's disease. Traditional pharmacology is poorly equipped to fix these errors. A pill cannot rewrite DNA. It can only manage the downstream symptoms, reducing inflammation or temporarily replacing a missing enzyme, while the faulty biological machinery continues to repeat the same mistakes. Gene therapy targets the internal machinery of the cell. It attempts to modify or replace the faulty genetic instructions at their origin, aiming to correct the biological output of the cell itself. This shift in focus turns the treatment of genetic disease into a question of mechanical access, finding ways to reach and repair the specific genes responsible for a patient's health. Out the virus, removing the elements that cause disease, and pack the empty shell with therapeutic DNA. These engineered delivery vehicles can then lock onto specific cells and inject their therapeutic payload exactly where it is needed. This specific technique is already working in the clinic. It is successfully being used to restore blood clotting proteins for patients with hemophilia and to reverse certain inherited forms of blindness. By mastering the mechanics of cellular infiltration, scientists bypass the pharmacy entirely. They compel a failing organ to manufacture its own internal medicine. Not all genetic conditions stem from a lack of production. Sometimes the threat comes from an overactive gene generating harmful, toxic proteins that build up in the body over time. In this animation, we see a technique called gene silencing, which utilizes a mechanism called RNA interference. Before the DNA's instructions can reach the cellular machinery that builds the toxic protein, a targeted suppression molecule intercepts and dissolves the messenger mid-route. This provides the toolkit with both a volume control and a kill switch, allowing for the precise suppression of toxic cellular factories. There are scenarios where simply bypassing or suppressing a mutation isn't enough. The DNA sequence itself has to be physically corrected. This brings us to the third tool, gene editing. The most prominent technology here is CRISPR-Cas9. Think of it as a set of programmable microscopic scissors. It scans the genome, locks onto a highly specific coordinate, and cleanly snaps the sequence in half so new, healthy code can be pasted in. Doctors are using this exact method to treat sickle cell disease. By physically cutting and rewriting the mutated sequences inside bone marrow cells, they restore the production of healthy, oxygen-carrying red blood cells. Looking at this flowchart, we can see how this editing process operates outside the body, known as ex vivo therapy. In treatments like CAR-T, doctors extract a patient's own white blood cells and take them to a lab to edit their DNA. Once they are armored with new receptors, the cells are infused back into the patient to actively hunt down cancer. Through gene editing, a patient's own biology becomes a custom-built, highly targeted weapon directed squarely at their specific disease. Manipulating the genome carries severe risks. If CRISPR misses its intended coordinate, it can accidentally sever healthy code, triggering new diseases. Delivery remains equally treacherous. Defensive antibodies will often swarm the site, shredding the delivery vehicles before the therapeutic DNA ever reaches its target. Even with these limitations, patients are currently receiving FDA-approved treatments for leukemia, muscular dystrophy, and several rare blood disorders. The pipeline is accelerating. Dozens of new targeted therapies are currently in late-stage clinical trials, poised to treat conditions that were entirely untouchable just a decade ago. As these tools refine, human genetics is moving away from being a permanent life sentence and toward becoming an editable draft. We are learning to rewrite the code we once thought was written in stone.